Hello everyone, this is Dr S S Bornare, Department of Genetics and Plant Breeding, KK College of Agriculture, Nasik. Today we are going to learn the topic hybrid seed production of rice. So let us see first the taxonomy of rice. The botanical name of rice is Oryza sativa, family Poaceae, and this rice is originated in Southeast Asia region and having chromosome number 2n is equal to 24, and the rice is predominantly a self-pollinated crop. And in case of rice, the commercial seed production or commercial hybrid seed production is generally done by using the cytoplasmic genetic male sterility system, that is Cgms system, and this may also known as three-line system, in which there are three lines: A line, then B line and R line. So this is the Cgms system in which there are three lines. This A line is female line and it is a male sterile line. Then this B line is male fertile line and also called as maintainer line. These A and B are isogenic lines except for the locus this sterility and fertility. This B lines is male fertile one, and the R line is restorer line and utilizes a male in hybrid seed production, and it is also a male fertile line. So by using this three line system, the hybrid seed is produced in case of rice, and the Cgms system is widely utilized in the hybrid seed production of rice. So the different seed produced in the different stages are: in breeder stage, the A and A into B line, B and R line are multiplied. In foundation seed, there is a cross between A into B and A R line are multiplied, and during the certified seed production, the A line, that is female line, which is male sterile, and R line, which is restorer line, utilized as a male, and by crossing these F1 seeds or F1 hybrid seed is produced. Then system of hybrid seed production. There are many systems available for hybrid seed production, out of which two are commonly utilized in hybrid seed production of rice, out of which only one, that is three-line system or Cgms system, is popular and widely used in hybrid seed production. And despite of that, two-line method or environmental genetic male sterility (EGMS) are also utilized for the hybrid seed production of the rice. This EGMS having another two types, that is PGMS, photosensitive genetic male sterility, and TGMS, that is thermosensitive, temperature sensitive and the light sensitive. These are the two types of genetic male sterility under the environmental genetic male sterility are utilized for the hybrid seed production. In the some part, the thermosensitive male sterility was developed in China and low temperature hilly areas of Tamil Nadu. So these are the two methods which are commonly utilized for hybrid seed production in the rice, out of which the three-line system, that is EBR line or Cgms system, is widely utilized and accepted. Then the genes for the fertility restoration in Cgms system is RF1 and RF2. Then commercial seed production technique, in which first is isolation. This isolation is achieved by many methods. First is space isolation, then time isolation and barrier isolation. In case of rice hybrid seed production, the space isolation is required. For foundation seed it is 20 meter, and for certified seed it is 100 meter. And if the space isolation is not possible, then we can isolate the field by using time isolation also. So the 20 days earlier, either earlier or later than the other varieties as compared to EMS line, compared with EMS lines. So these male sterile lines are sold earlier or later than by the difference of 20 days than other varieties. So this is the time isolation. Then next is barrier isolation. If these two are not possible, then we can isolate our hybrid seed production plot by barrier isolation also, in which a 30-meter of wood lot or tall crops can grow around our hybrid seed plot. Then plastic sheets of 2-meter height are also placed outside the border for the entry of to block the entry of the foreign pollen, that is a barrier isolation. So these are the different types of isolation by which we can isolate our field from the foreign pollen. So season, so rice is grown in Kharif and Rabi season also. In Kharif, May or June is the sowing timing, and in Rabi, December and January is the sowing timing. And Rabi is more suitable than Kharif due to the problem of the rainy season. The Rabi season rice or hybrid seed production is more suitable as compared to Kharif. So, what are the favorable climates or climatic conditions during the flowering for our higher seed set in hybrid seed production? So, let us see the point, in which first is a daily mean temperature of a range 24 to 30 degrees Celsius. So this is the good climatic conditions for the higher seed set. The relative humidity is in between 70 to 80%, and the difference between day and night temperature, that is also called as diurnal variations, should be 8 to 10 degrees Celsius, and sufficient sunshine and moderate wind velocity of 2 to 3 meter per second. So these are the different climatic factors which are found to be good for higher seed set. Then free from continuous rain for above the 10 days during the peak flowering session. So this is the another one major climatic conditions which during the Kharif season a limiting factor. That means during the Kharif season the continuous rain causing the problem in higher seed set. So for that efficient seed set, the continuous rain should not be there during the peak flowering season. Then seed set and seed yield will be affected if temperature is below 20 degrees Celsius. So this is the temperature range beyond which or below which the seed set is highly hampered, so that the range of the temperature is not gone below the 20 degrees Celsius and above 35 degrees Celsius during the time of flowering, and it does not exceed 35 degrees Celsius also. Means in between 20 to 35 degrees Celsius it is good for higher seed set during the flowering. Then seed rate in case of female line it is 20 kg per hectare, and for male line it is 10 kg per hectare. Nursery management, how the nursery is managed: keep irrigation channels separately for the parental lines. For both parental line, that is male and female, there must be a separate irrigation channel. Then for December to January sowing, take up a stagger sowing for male twice or thrice. So in case of hybrid cell production, there is a problem of synchronization in male and female parent, which is prerequisite for higher hybrid seed set. So for that, the stagger sowing of male twice or thrice is essential. Then with the twice or thrice with the interval of 10 to 15 days, that is 3, 10 and 15 days for effective seed setting. So this is the practice required to achieve the synchronous flowering in male and female line. Then keep the nursery area free from weeds. Then apply diammonium phosphate at the rate of 2 kg as basal to get a vigorous seedling. For April or May sowing, sow the male 5 and 10 days after female line. So this is again one, the synchronous, this is practice is done for synchronous flowering in male and female parent. Even split application of the fertilizer and nitrogen is favorable for production of vigorous seedling. So these are the some key points during the nursery management. Then main field transplanting. For the transplanting spacing, the spacing between A lines is 15 into 15 centimeter. Between A and R line, it is 20 into 15 centimeter, between R line 30 into 15 centimeter, and age of the seedling at the transplanting, in case of A line it is 25 days, and in case of R line it is 14, 18, 20 days. So these are the age of seedling at the time of transplanting. And the fertilizer and manures for hybrids, 150, 60, 60 NPK kg per hectares are required for the hybrid seed production, and N and K in 3 splits, that is first during basal, and second active tailoring, and third during panicle initiation. So this is the fertilizer requirement. Then staggered sowing for parents for synchronization. So the synchronization of male and female parent is a key factor for higher seed set or higher seed set in the hybrid seed production. So as the seed set on the cytoplasmic male sterile line depends on the cross pollination, it is mostly important to synchronize the heading date of the male and female parents, especially for the hybrid combinations having parents with quite different growth duration. So there is a requirement of synchronization in the male and female parents flowering dates. For that there is a mechanism called as a stagger sowing is done, and that's why the heading date of male and female parents are matched. And in addition, in order to extend the pollen supply time, the male parent is usually seeded twice or thrice an interval of 4 to 5 days. So this is the another practice that ensure the extend the pollen supply for long duration, so that the male parent is usually seeded twice or thrice at an interval of 4 to 5 days, so that ample quantity of pollens or pollen load is there available and for long duration also. Then a row ratio, there is 8 to 2 or 10 to 2 female as to male ratio in case of hybrid seed production. That means 8 female line and 2 male line in between. So this is the row ratio or planting ratio in case of hybrid seed production of rice. Then what are the factors that influencing the row ratio, in which the plant height of the pollinator, growth and vigor of the pollinator, that is male plant, then size of the panicle and amount of the residual pollen, then duration and angle of floret opening in cytoplasmic sterile line, and stigma exertion of CMS lines. So these are some key factors that decide the row ratio or that affect the row ratio. So while maintaining the row ratio, we have to take consideration of these points, and according to which the row ratio is decided as 8:1 or 8:2 or 10:2 of female and male line. We can see here in diagram also the row ratio of the male female line. Then besides the normal pollination, there are supplementary pollination is needed in some cases. The natural outcrossing was recorded less than 10% by Ramalingan and co-workers in 1994. So according to their study, the natural outcrossing was recorded less than 10%, but this depends upon the wind direction and velocity of the wind in the particular area. So for that, the shaking of R-line panicles by rope pulling at the panicle level or rod driving during the anthesis can make their anther dehiscence and spread the pollen widely and evenly. Thus, the outcrossing rate could be increased, and this is nothing but the supplementary pollination. Means besides the natural one, we supplement the pollination by rope pulling or rope pulling at panicle level or rod driving during this anthesis. It is called as a supplementary pollination, and it is more effective especially on the calm or breezy days. Generally, the supplementary pollination is carried out at the 30 minutes interval for 5 times daily, both morning and evening, during the peak anthesis time, that is 10 to 12 am and 2 to 4 pm, until the no pollen remain on the R line, and it is not needed when the wind is greater than the moderate breeze. So this supplementary pollination is needed when the there is a calm or less wind is there. So in that case, the supplementary pollination is carried out 30 minutes interval for 5 times during the morning and evening, and thus there is no pollen remain on R line, and this pollen is spread over the A line. In the diagram we can see the supplementary pollination is practiced. Then foliar spray of 2% DAP increases the yield and qualities of seed, and for that, in case of short duration variety, first spray of 60 days after the sowing, and second 80 days after the sowing. Then in case of medium duration, first spray is given on the 80 days after sowing, and second is on 100 days after sowing. Next important is rogging in the hybrids. Rogging is essential to maintain the genetic purity of the hybrid seed, and rogging is done in case of hybrid in A line to remove the pollen shaders. And in A line, only 40 to 50% of the seed set is possible, as it is the male sterile line. The pollen from the pollen shader line is transferred to this line, and only 40 to 50% of the seed set is possible in A line. If it is greater than 60 to 70% seed is noticed, the panicle is drooping, it would be R line or other varieties. So we have to rogue out or remove out these plants. Here a table showing the different types of plants or rogue which is to be removed in the disease plant, then parental lines and early flowering plant. So in case of disease plant, all the disease plant in all lines must be rogued. Then in case of early flowering or late flowering also, all the lines having all the plants which are early or late to the normal line must be to be removed. And in case of parental line, in A line, R line and B line must be removed. In B line, A line and R line, and in R line, B line and A line must be removed or rogued out. Then last one is harvesting, threshing and drying. So turning up 90% of green seeds to the straw yellow color, this is the physiological maturity sign. At this physiological maturity stage, the harvesting is done just after this physiological maturity, and when the moisture content is be 17 to 20%, the male parent should be harvested first, and the care should be taken to avoid the admixture of male line and female line while harvesting. So during harvesting we have to take care of male and female line so that they do not mix it with each other. The female parent should be threshed at the 16 to 17% moisture percent, and in separately and in a well threshing floor, so that there is no physical mixture or admixture of any other lines with the female line. The threshed seed should be winnowed and dried to reduce the seed moisture content at the 12%. While drying, seed should not be dried under the direct sun between 12 to 3 p.m. during the hot sunny days. So these are the some techniques or precautions we have to take care while harvesting, threshing and drying of the hybrid seed. Last is storage. For short-term storage, we use generally gunny bags or cloth bags, and for long-term storage, use polythene bag of more than 700 gauge and dry the seed 8% moisture content. When compared with the other varieties, the hybrids and parental lines, A and B lines are poor in storability. Their storability orders are given here. The order of storage potential is greater in R line, then F1, then B and then A. And lastly, the seed yield, hybrid seed yield or F1 hybrid seed yield is 800 to 1200 kg per hectare. So thanks. This video is for educational purpose only and references are as below. Thanks once again.